Hey, sleepers! Tonight, we're going on a journey into the very code of life, a place of quiet humming and intricate secrets. Imagine a library, endless and vast, holding the stories of every living thing. The air is still, and the only sound is the gentle turning of pages, each one a blueprint for existence. We are going to find a book in this library, the one labeled human, and we are going to look closer than ever before at the very ink on the page. We will find that some of the words were not written by us, but by a visitor from long ago. So before you get comfortable, take a moment to like the video and subscribe. It helps our little corner of the internet grow, and we are so grateful to have you with us tonight. Feel free to share where you're listening from in the comments below, and what time it is in your part of the world. It's a wonderful way to feel connected, knowing we are all settling down under different skies. So close your eyes, take a deep breath, and get ready to drift off as we explore the virus that rewrites your DNA. Let's begin our journey with a simple thought. Imagine a tiny, tiny messenger. This messenger is so small, you could fit billions of them on the head of a pin. It is not truly alive, not in the way a flower or a bee is alive. It cannot grow or eat or make its own energy. It is just a package, a neat little bundle of information with a single urgent purpose: to make more of itself. This is a virus. Now, most messengers you might think of carry a note, deliver it, and leave. They might knock on the door of a cell, hand over their message, and the cell reads it and does what the note says. But the messenger we're talking about tonight is different. It's a special kind of messenger, a retrovirus. It's a bit of a trickster, a master of disguise and infiltration. Inside your body, your cells have a master library. This library is called the nucleus. And inside this library is the most important book in your entire existence. It's your genome, written in the language of DNA. This DNA is like a set of incredibly detailed, permanent blueprints. It's made of two long, intertwined strands, a double helix, like a beautiful spiral staircase. This book contains every instruction needed to build you and keep you running. Every cell in your body has a copy of this book. The language is permanent, stable, and carefully protected. Our special messenger, the retrovirus, doesn't carry its instructions in the permanent language of DNA. It carries them in a different language, a more temporary one called RNA. You can think of RNA as a spoken message or a quick note jotted on a piece of paper. It's a single strand, less stable, and usually meant for short-term tasks. In your cells, RNA is used all the time to carry copies of instructions from the main DNA library out into the cell to build things like proteins. It's the daily memo, not the permanent archive. So here comes our retrovirus, floating along until it bumps into one of your cells. Let's say it's a helper T cell, a guardian of your immune system. The virus has a special key that lets it unlock the door to this cell. It slips inside, shedding its outer coat and releasing its contents. And here's where the magic or the mischief begins. The virus has brought along its own special tool. It's an enzyme, a tiny biological machine called reverse transcriptase. The name itself gives away its secret. Normally, in your cells, the flow of information is from the permanent DNA to the temporary RNA. This is called transcription. But this virus does it backwards. It performs reverse transcription. Using this magical enzyme, the virus takes its temporary RNA message and rewrites it into the permanent language of DNA. It's like hearing a story whispered in the air and having the power to instantly etch it onto a stone tablet. The flimsy, single-stranded RNA becomes a sturdy, double-stranded piece of DNA. This newly made viral DNA is now a perfect counterfeit of the cell's own genetic material. It speaks the same language. It looks the part. It's ready for the next step of its infiltration. Now the viral DNA, this little piece of stone tablet, needs to find the main library, the nucleus. It travels to the heart of the cell, where the master blueprint, your chromosome, is kept. And here, the virus uses its second secret weapon. It's another enzyme, another tiny machine, called integrase. Think of integrase as a master locksmith with a tube of superglue. It finds a spot in your long, spiraling DNA, your chromosome. It makes a tiny, precise snip, opening up your blueprint. And then it pastes the viral DNA right into that gap. It glues it in, sealing the seams perfectly. The cell's own repair machinery comes along and tidies up, making the insertion seamless. And just like that, it's done. The viral DNA is now a permanent part of your cell's own DNA. It is no longer a visitor. It is a resident. This integrated viral DNA has a special name. It is now called a provirus. It's a secret chapter, silently inserted into the great book of you. From this moment on, something profound has changed. Every time this cell divides, it first has to copy its entire library of DNA. And as it copies its own genes, its own instructions, it will also unknowingly copy the viral genes. The provirus is replicated right along with everything else. The secret chapter is copied into every new book. If this cell divides into two, both daughter cells will have the viral DNA. If they divide into four, all four will have it. The infection is now permanent in this cell and all of its descendants. It cannot be washed out. It cannot be removed. It is part of the code. The human immunodeficiency virus, or HIV, is the most famous example of this process. It targets those crucial immune cells, the CD4 T cells. By inserting its DNA into these cells, HIV establishes a permanent, lifelong reservoir in a person's body. The provirus can lie dormant, silent, for years. A hidden chapter, waiting. Or it can be activated, and the cell will begin reading the viral instructions. It will start to produce the parts for new viruses. The cell becomes a factory, churning out thousands of new viral messengers, which then go on to infect other cells. This is how the virus spreads within the body, slowly and methodically rewriting the story of the immune system, one cell at a time. It's a quiet, cellular heist of incredible elegance. And it all hinges on that one audacious act, rewriting the master code itself. The discovery of this incredible process was a revolution in biology. For a long time, the central rule of molecular biology, what was called the central dogma, was considered absolute. Information flows in one direction. It flows from the permanent DNA library to the temporary RNA message, and then from the RNA message to the creation of a protein. DNA to RNA to protein. It was a one-way street. The idea that information could flow backwards, from RNA back into DNA, was considered heresy. It was like saying you could unbake a cake, or that a spoken word could magically write itself into a book. But in the 1960s, a scientist named Howard Temin began to suspect that this dogma wasn't the whole story. He was studying a virus that caused tumors in chickens, a retrovirus, though the term wasn't widely used yet. He noticed that the virus seemed to permanently change the cells it infected. Even after the initial infection was gone, the cells and their descendants continued to behave differently, as if they had a new, lasting instruction. Temin proposed a wild idea. What if the virus was somehow inserting its RNA-based genetic information into the DNA of the chicken cells? For years, his idea was met with skepticism, even ridicule. The scientific community was firm in its belief of the one-way street, but Temin persevered, convinced by his observations. He spent the better part of a decade trying to find the mechanism, the tool that could perform this reverse flow of information. Then, in 1970, a breakthrough. Two scientists, working completely independently in their own labs, found the proof. Howard Temin, at the University of Wisconsin, and another brilliant scientist named David Baltimore at MIT both discovered the enzyme responsible. They both isolated and identified reverse transcriptase, the molecular machine that could read RNA and write DNA. It was a stunning confirmation of Temin's long-held theory. The discovery shattered the old dogma and opened up an entirely new field of biology. It showed that the flow of genetic information was more complex, more fluid than anyone had imagined. For this monumental discovery, Temin and Baltimore, along with another researcher named Renato Del Becco for his work on tumor viruses, were awarded the Nobel Prize in Physiology or Medicine in 1975. This discovery was a key that unlocked a huge mystery, but it took a few more years for its full implications for human health to be realized. For a while, these retroviruses were mostly known in animals. It wasn't until 1980 that the first human retrovirus was identified. A team led by Dr. Robert Gallo at the National Cancer Institute found a virus they called human T lymphotropic virus 1, or HTLV1. They discovered it was the cause of a specific rare type of leukemia. This was the first time a retrovirus was definitively linked to a human disease. Then came the 1980s, and with it, a terrifying new epidemic. A mysterious illness was causing the immune systems of healthy young people to collapse. No one knew what caused it. The race was on to find the culprit. In 1983 and 1984, research teams, including Gallo's in the United States, and a team led by Luc Montagnier and Françoise Barré-Sinoussi in France, identified the virus responsible for AIDS. They found that it was a retrovirus. And suddenly, everything clicked into place. The discovery of reverse transcriptase 13 years earlier now explained the insidious nature of this new disease. It explained why the infection was permanent. The virus wasn't just attacking the body. It was writing itself into the body's very instruction manual. It was hiding in the DNA. This is where we must also mention the pivotal work of Dr. Flossie Wongstow, a brilliant virologist and molecular biologist who worked alongside Robert Gallo. She was the first person to clone HIV. By doing this, she was able to map out its genetic structure. This work was absolutely essential. It allowed for the development of the first blood tests for HIV, which could detect the virus's genetic material. Dr. Wongstow, a woman of Chinese descent who became a leading figure in a male-dominated field, provided the genetic map that has guided AIDS research ever since. Her contribution highlights how science is a collaborative effort, built upon the work of many brilliant minds from all backgrounds. So, The history of retroviruses is a story of scientific rebellion, of challenging a central belief and being proven right. It's a story of how a fundamental discovery about how life works provided the crucial key to understanding a devastating new human disease. It showed us that sometimes, to find the truth, you have to be willing to believe that information can, indeed, flow backwards. Now, this idea of a virus writing itself into our DNA might seem like a modern phenomenon, a consequence of our encounters with new pathogens, like HIV. But what if I told you that this process is ancient? What if I told you that this isn't just a story about individual infections, but a story about the evolution of our entire species? Take a moment and just feel the gentle weight of your body, the quiet rhythm of your breath. Within every single one of your cells, you are carrying the ghosts of infections past. Let's go back to our library analogy. We know that a virus like HIV can insert a new page into the book of a single cell. But what if that infected cell wasn't a skin cell, or a lung cell, or an immune cell? What if the cell that got infected millions of years ago in one of our distant, furry ancestors was a germline cell, a sperm cell, or an egg cell? If a retrovirus writes its code into the DNA of a germline cell, something remarkable happens. That viral code, the provirus, is no longer just in that one individual. It's in the blueprint that will be passed on to the next generation. The offspring that develops from that egg or sperm will have the viral DNA in every single cell of its body. And when that offspring grows up and has its own children, it will pass that same viral DNA on. It becomes a permanent part of the family line, inherited just like the gene for eye color or the gene for curly hair. This has happened. It has happened many, many times over the course of mammalian evolution. Today, when scientists analyze the human genome, that 3 billion letter instruction book, they find something astonishing. Approximately 8% of your entire DNA is made up of these ancient retroviral sequences. Let that sink in. 8%. The part of your DNA that contains the actual recipes for proteins, the genes that build your body, makes up only about 2%. This means you have 4 times more viral DNA and eerie genome than you have human protein-coding genes. These ancient viral fragments are called endogenous retroviruses, or ERVs. Endogenous just means coming from within. They are now part of us. Most of these ERVs inserted themselves into our ancestral genomes tens of millions of years ago. We can date them by comparing the genomes of different species. For example, if we find the exact same viral insertion in the exact same spot in the DNA of humans, chimpanzees, and gorillas, we know that the infection must have happened in a common ancestor before our species split apart. It's a form of genetic archaeology. We are digging through our own DNA and finding the fossilized remains of ancient viruses. There are hundreds of thousands of these viral fragments scattered throughout our chromosomes. They are like ghosts in the machine. Most of them are no longer functional. Over millions of years, they have accumulated mutations, typos in their genetic code. The ink has faded. The instructions are garbled. They can no longer produce new viruses. They are silent relics, broken pieces of ancient viral machinery. They are the genetic fossils of pandemics our distant ancestors survived. Imagine walking through a desert and finding the fossil of a seashell. It tells you that millions of years ago, that desert was once a vibrant ocean. In the same way, these ERVs in our DNA fossils, that tell us about the ancient viral oceans our ancestors swam through. They are a permanent record of our evolutionary past, a testament to a long and intimate relationship between viruses and our own lineage. So as you rest here, just be aware of this deep history within you. Your DNA is not just a blueprint for you, the individual. It is an archive, a history book, containing the stories of countless generations and the echoes of a viral world that existed long before we did. This 8% of you is a direct, physical link to the deep, deep past. It's easy to think of this viral DNA as junk, as clutter, ancient garbage left over from forgotten battles. For a long time, that's what scientists thought. This junk DNA, including the vast graveyards of endogenous retroviruses, was considered meaningless, inactive filler material. But nature is rarely so wasteful. In recent years, we've begun to realize that some of these ancient viral ghosts are not sleeping as soundly as we thought. In fact, some of them are not ghosts at all. They have been repurposed. They have been given new jobs. And some of these jobs are absolutely essential to what it means to be human. This is perhaps the most mind-bending part of our story tonight. The virus, the invader, the thing we think of as other, has, in some cases, become an indispensable part of us. Let's talk about one of the most miraculous processes in all of biology, the formation of the human placenta. The placenta is a remarkable temporary organ that forms during pregnancy. It is the life support system for a developing baby, connecting it to the mother. It provides oxygen and nutrients, and it removes waste. It's a bridge between two genetically distinct individuals. For this bridge to work, a very special layer of cells has to form. This layer, called the syncytial trophoblasts, is on the outer surface of the placenta, directly in contact with the mother's tissues. This layer is bizarre. It's not made of individual cells neatly arranged side by side. Instead, it is a single, giant, continuous cell that contains thousands of nuclei, all sharing the same cytoplasm. It's a massive fusion of cells. This structure forms a perfect barrier. It's a fortress wall that protects the fetus from the mother's immune system, which might otherwise recognize the fetus as foreign and attack it. It also allows for the incredibly efficient transfer of nutrients. How do all these individual cells fuse together to become one giant supercell? They need a special kind of protein, a fusogen, a molecule that can merge cell membranes. For years, the origin of this protein was a mystery, and then scientists found it. It's a protein called syncytin, and when they looked at the gene that codes for syncytin, they found it sitting right in the middle of an ancient, crippled endogenous retrovirus. The syncytin gene is a repurposed viral gene. Specifically, it comes from what used to be the envelope gene of a retrovirus. The envelope gene is what a virus uses to build the protein on its surface, the key that it uses to latch onto a cell and fuse with it to get inside. Our ancestors were infected by this virus millions of years ago. The virus wrote itself into our germline. And over time, most of the virus was silenced and mutated away. But our bodies kept this one useful gene. We co-opted the virus's own tool for breaking and entering, and we turned it into a tool for building and nurturing. We took the gene that allowed a virus to fuse with a cell, and we used it to fuse our own cells together to build the placenta. It's so essential that if you disable the syncytin gene in mice, they cannot form a proper placenta, and the embryos do not survive. And it's not just one gene. We found a second similar gene, called syncytin 2, which also came from a completely different retroviral infection at another point in our history. So this trick was so good, evolution borrowed it twice. Take a moment to let that sink in. The creation of new human life, the very first connection between a mother and a child, is made possible by a protein that we inherited from an ancient virus. Without these long-dead viruses, mammalian pregnancy as we know it might not exist. Every person who has ever been born owes a debt of gratitude to these viral ghosts in our genome. The line between host and parasite, between friend and foe, completely dissolves. And the story doesn't end there. These ERVs do more than just build placentas. They also act as switches that control our own genes. Remember, a provirus inserts itself into our DNA. Where it lands can have a big effect. Sometimes, a virus inserts itself near a human gene. The virus's own control sequences, the parts that say, "turn me on," can sometimes start affecting the nearby human gene. They can act like a new dimmer switch, turning a gene on in a new tissue, or at a new time during development. Scientists have found that many ERVs are highly active during early embryonic development. They switch on right after fertilization, and then are silenced again as the embryo develops. Their activity seems to be crucial for orchestrating the complex dance of gene expression that turns a single cell into a complex organism. They help to tell genes when and where to turn on, shaping the very earliest stages of our existence. Some of these ERVs are even thought to play a role in maintaining the pluripotency of stem cells, their magical ability to become any type of cell in the body. Even more fascinating, some of these ERVs may still protect us. One family of human endogenous retroviruses, called HERV-K, is very active in early embryos. Some researchers believe that the expression of these viral proteins might create a state of viral immunity, making the embryo resistant to infection by modern external viruses. The ghost of an old virus stands guard, protecting the new life from its more aggressive relatives. It's a beautiful, poetic irony. The ancient invader has become the guardian of the gate. So the next time you think of your DNA as a pure, pristine blueprint, remember these viral collaborators. They are not just junk. They are architects and regulators and protectors. They are a fundamental part of our biological story, woven so deeply into our fabric that we can no longer tell where the virus ends and we begin. The impact of these viral insertions goes beyond building specific body parts or regulating a few genes. It's bigger than that. Retroviruses have been a powerful engine of evolution, a constant source of novelty and change that has helped to shape the entire trajectory of mammalian life. Evolution works with what it has. It tinkers. It modifies. It needs raw material to work with, and that raw material is genetic variation. Mutations, small changes in the DNA sequence, are one source of variation. But a retrovirus inserting itself into a genome is a huge change. It's not just changing one letter. It's adding an entire new chapter, thousands of letters long. Imagine our genome as a landscape. For millions of years, it's been shaped by wind and rain, the slow, gradual forces of mutation. But a retroviral insertion is like a meteorite strike. It can dramatically reshape the landscape in an instant. Sometimes the impact is catastrophic. If a virus lands in the middle of a vital gene, it can disable it, potentially causing disease or death. These insertions are quickly weeded out by natural selection. But sometimes the meteorite lands in a place where it does something interesting. it doesn't cause a disaster. Instead, it creates a new mountain or carves out a new river valley. We've already seen how these insertions can provide new proteins, like syncytin. But perhaps their most powerful role is as those genetic dimmer switches, the regulatory elements. A virus comes with its own set of instructions for when and how it should be activated. These instructions are called promoters and enhancers. When a virus inserts itself into our DNA, it brings these control panels with it. Suddenly, a human gene that was sitting nearby might find itself next to a powerful new switch. Maybe this switch turns the gene on in the brain for the first time. Or maybe it turns it on only in response to stress. This can create new traits. This process, where mobile genetic elements like ERVs rewire the gene regulatory networks of a host, is now seen as a major driving force in evolution. It's a way to quickly create new patterns of gene expression, which can then be tested by natural selection. It has contributed to the differences we see between species. Part of what makes a human different from a chimpanzee isn't just the small differences in our protein-coding genes, but the vast differences in how those genes are regulated. And many of those regulatory differences have been scattered throughout our genome by ancient viruses. There is one example of this that is particularly poetic and profound. It has to do with something very close to our hearts and our minds' memory. The ability to form long-term memories, to learn from experience and build a sense of self, relies on a process called synaptic plasticity. This is the ability of the connections between our neurons, our brain cells, to strengthen or weaken over time. It's how learning is physically encoded in the brain. A few years ago, scientists were studying a gene that is absolutely critical for this process. The gene is called ARC, which stands for Activity-Regulated Cytoskeleton-Associated Protein. When a neuron is active, the ARC gene is switched on, and it helps to remodel the synapse, strengthening that connection. Without ARC, long-term memory is impossible. When scientists looked closely at the ARC gene, they noticed something familiar. It looked suspiciously like a gene from a retrovirus. Specifically, it looked like the viral gene called GAG, which viruses use to build their structural capsule, the very package that holds their genetic material. The evidence is now very strong that our essential memory gene, ARC, originated from an ancient retroviral-like element that inserted itself into the genome of a four-limbed vertebrate ancestor some 400 million years ago. What's more, the ARC protein behaves in a shockingly virus-like way. It actually assembles itself into little capsules, just like a virus does. It then packages up RNA messages inside these capsules and sends them from one neuron to another across the synapse. It is, for all intents and purposes, a domesticated virus, repurposed by our own neurons to communicate with each other. The very mechanism our brains use to store information, to create memory, appears to be a repurposed piece of viral machinery. The ability to remember your first love, or the smell of your childhood home, or the words of this story, is powered by the ghost of a virus. It's an evolutionary plot twist of the highest order. The invader didn't just change our bodies. It may have given us the very tools we use to build our minds. While many of these ancient viral residents are silent, broken relics, not all of them are. Some of the younger ones are still capable of waking up. It's like having an old, dusty machine in the basement. Most of the time it sits there, quiet and harmless. But under certain circumstances, a power surge, a jolt, a bit of spilled water, it might roar back to life, at least partially. The youngest family of human endogenous retroviruses is called HERV-K. Some of these insertions are so recent that they are not shared by all humans. They happened after our lineage split from chimpanzees, and some even happened after modern humans diverged from Neanderthals. These HERV-K elements are the most intact. They have the fewest mutations. While none of them seem to be able to create a fully infectious, complete virus anymore, some of them can still produce viral proteins. Our bodies have sophisticated defense mechanisms to keep these elements silenced. We use a process called methylation, which is like putting chemical locks on the DNA, preventing it from being read. But sometimes these locks can fail. In certain disease states, these ancient viruses can be reactivated. This aberrant activation has been linked to several complex diseases. For example, in autoimmune diseases like multiple sclerosis and lupus, the body's immune system mistakenly attacks its own tissues. Researchers have found that in patients with these conditions, certain HERVs are switched on at abnormally high levels. One theory is that the production of these viral proteins might confuse the immune system. The immune system sees these strange, foreign-looking proteins and launches an attack. But because these proteins are being produced by our own cells, the attack is directed at ourselves. The ancient enemy within might be acting as a fifth colonist, tricking our own defenses into starting a civil war. There's also a strong link between EREs and some types of cancer. Cancer is a disease of uncontrolled cell growth, often caused by mutations that mess up the normal rules of cell division. In many tumors, the normal silencing mechanisms that keep ERVs locked down are broken. As a result, these viral sequences can become reactivated. It's not always clear if this is a cause of the cancer or just a symptom of the general chaos inside a cancer cell. But in some cases, it's thought that the reawakened viral proteins might help the tumor to grow or to evade the immune system. The virus's ancient programming, which is all about replication and survival, could be giving the cancer cell an advantage. It's important to approach this with a calm and measured perspective. This isn't something to be anxious about. For the vast majority of people, these ERVs remain silent and harmless, locked away by a billion years of evolved defenses. The connection to disease is an area of intense research, and it's very complex. It's not as simple as this virus causes this disease. It's more about a delicate balance. When the body's own regulatory systems are weakened or disrupted, these old programs can start to run again, sometimes causing glitches. It reminds us that our genome is not a static blueprint, but a dynamic, living ecosystem with its own internal conflicts and alliances, a history of ancient struggles that can sometimes echo into the present day. Let's bring our focus back to the here and now, to the retroviruses that are actively infecting people today. We've talked about HIV, but there's much to learn from how it and other modern retroviruses behave inside our cells. A key question has always been, when a retrovirus integrates into our DNA, where does it go? Is it completely random, like throwing a dart at a giant encyclopedia in the dark? Or does it have preferences? It turns out, integration is not random at all. Different retroviruses have different tastes for where they like to set up shop. This choice of real estate has huge consequences for the infected cell and for the person. HIV, for instance, shows a strong preference for integrating right into the middle of active genes. These are the parts of the DNA that are open for business, unwound and busy, being transcribed into RNA. From the virus's point of view, this makes perfect sense. An active gene is a place where there's lots of machinery, a bustling hub of activity. It's a good place to be if you want your own genes to be read and turned into new viruses. It's like setting up your hot dog stand in the middle of Times Square instead of a quiet suburban street. However, this preference can sometimes be a problem for the host cell. Integrating into a gene can disrupt it, potentially damaging the cell. But for the most part, HIV's integration pattern is about maximizing its own replication, not about causing other specific diseases. Another human retrovirus, HTLV1, the one discovered by Robert Gallo's team, has a different strategy. It doesn't seem to care so much about being right in the middle of a gene. Instead, it has a tendency to integrate near specific types of regulatory regions, particularly those that control cell growth. This is where its link to cancer comes from. In a small percentage of people infected with HTLV1, the virus, by chance, inserts its DNA near a powerful gene that tells a cell to divide. The virus's own on switch, its promoter, then acts on this nearby human gene, essentially locking the accelerator pedal to the floor. The T cell begins to divide uncontrollably, leading to adult T cell leukemia. It's a tragic lottery, a case of the virus landing in exactly the wrong place at the wrong time. The choice of integration site is a complex dance between viral proteins and our own cellular proteins. The virus's integrase enzyme doesn't work alone. It partners with proteins inside our cells that guide it to certain regions of our chromosomes. Different viruses use different cellular partners, which is why they end up in different neighborhoods of the genome. What this means is that in a single person with a chronic retroviral infection like HIV, there isn't just one provirus. There are thousands, millions of infected cells, and in each one, the virus might be integrated in a slightly different place. This creates a vast and unique mosaic of integration sites. If you were to map out all the proviruses in one person's body, you would get a unique genetic fingerprint of their infection. This diversity is one of the things that makes these infections so challenging. Even if you could find a drug to target the virus in one location, there are thousands of other copies hiding in other parts of the genome, ready to reactivate. It's a distributed network, a decentralized enemy, woven into the very fabric of the self. We have talked about how this process of a virus becoming a permanent part of the species' DNA happened, our ancestors millions of years ago. It can feel like an abstract ancient story. But what if we could watch it happening right now? What if there was an animal population on Earth today that is in the middle of this very process? There is, and they are the sleepy, eucalyptus-munching koalas of Australia. Koalas are currently being invaded by a retrovirus called, appropriately, koala retrovirus, or CORV. And it is providing scientists with a remarkable real-time window into evolution. It's like having a time machine that lets us watch how our own ERVs got there. The situation with CORV is fascinating because it exists in two different forms in the koala population. In some parts of Australia, particularly in the northern states like Queensland, KORV is fully endogenous. That means every single koala has the virus integrated into its germline DNA. Every cell in their body contains CORV, and they pass it down from parent to joey. For these koalas, the virus is already a part of who they are, a part of their genome. The invasion is complete. But in other parts of Australia, like in the south and on islands like Kangaroo Island, the koalas were, until recently, free of the virus. Now, KORV is spreading through these southern populations as a contagious infection, just like any other virus. It is an exogenous infection, one that comes from the outside. Some koalas get sick, and some don't. But the virus is spreading. What scientists are witnessing is the virus making the leap. In these southern populations, they are finding some koalas where the virus has successfully infected their sperm or egg cells. These koalas are the bridge. They are the individuals in whom a contagious disease is transforming into an inheritable genetic trait. Their offspring will be the first in their lineage to be born with KORV as a permanent part of their DNA. We are literally watching the endogenization of a retrovirus. This provides an incredible living laboratory. We can study which koalas are more susceptible. We can see where the virus tends to integrate in the koala genome. We can study the effects of these new insertions. In the northern koalas, where the virus has been endogenous for longer, we can see the beginnings of how the host adapts. The koala genome is already starting to silence and control the virus, just as our ancestors did. We can see the first steps in the long, slow process of taming the invader and potentially, one day, repurposing it. The koala story is a powerful reminder that evolution is not just something that happened in the distant past. It is an ongoing, dynamic process. The intimate dance between viruses and their hosts is still happening, still shaping the genomes of life on Earth. The genetic book of life is still being written, and sometimes the viruses are still holding the pen. It takes a story that feels ancient and abstract and makes it immediate, poignant, and observable in the lifetime of a single furry marsupial. It might seem like this story is all about the power of the virus. The virus invades, it rewrites, it becomes a part of us. But the story of science is a story of turning knowledge into power. Once we understand a mechanism, we can often harness it. And that is exactly what has happened with retroviruses. We have taken the virus's most dangerous weapon, its ability to permanently write genes into our DNA, and we have turned it into one of the most promising tools in modern medicine. This field is called gene therapy. The idea is simple and beautiful. Many genetic diseases are caused by a single faulty gene. A person is born with a typo in their DNA that prevents them from making a crucial protein. What if you could deliver a correct, working copy of that gene into their cells? You wouldn't even need to fix the original typo. You just need to add a new, functional instruction. But how do you deliver that gene? You need a vehicle, something that is an expert at getting inside cells and inserting genetic material. What better vehicle than a retrovirus? An entity that has been perfecting this exact job for hundreds of millions of years. So, scientists became bioengineers. They learned how to take a retrovirus, like a lentivirus, which is the family that HIV belongs to, and retool it. They carefully remove all the viral genes that cause disease. They take out the instructions for making new viruses. They strip it down to its essential chassis, the ability to enter a cell and integrate a piece of genetic code. Then, in the place where the harmful viral genes used to be, they insert their therapeutic cargo, a healthy human gene. The result is a modified, disarmed viral vector. It's like taking a delivery drone, removing its surveillance camera, and replacing it with a package containing life-saving medicine. This vector can infect a cell, but it can't replicate. All it can do is deliver its package and permanently integrate that healthy gene into the cell's DNA. One of the most spectacular successes of this approach is a treatment called CAR T cell therapy, which is revolutionizing cancer treatment. The process is incredible. Doctors take blood from a cancer patient and isolate their T cells, their own immune soldiers. In a lab, they use a modified retroviral vector to deliver a new gene into these T cells. This new gene codes for a special receptor, a chimeric antigen receptor, or CAR. This new receptor acts like a highly specific targeting system, teaching the T cell to recognize and viciously attack the patient's cancer cells. After these T cells have been reprogrammed, they are grown into an army of millions in the lab and then infused back into the patient. This army of supercharged, cancer-hunting T cells then goes to work, seeking and destroying the tumor. It is a living drug, and it has led to remarkable, long-lasting remissions in patients with certain types of leukemia and lymphoma who had run out of all other options. We are using a virus's own trick to teach our immune system how to be a better fighter. This technology is also being used to treat inherited genetic diseases. For devastating conditions like sickle cell disease or beta thalassemia, where a person has a faulty gene for hemoglobin, doctors can take out a patient's own blood stem cells, use a retroviral vector to give them a working copy of the hemoglobin gene, and then return the corrected cells to the patient. These corrected stem cells can then produce healthy red blood cells for the rest of the person's life. Of course, it's not without risks. The early days of gene therapy had some serious setbacks. Because these vectors integrate into the DNA, there is always a small risk that they could land in a bad spot and, like HTLV1, accidentally turn on a cancer-causing gene. But our understanding has grown immensely. We now design safer vectors that have a lower chance of doing this. And by understanding the integration preferences of different viruses, we can choose the safest possible delivery vehicle for the job. It's a field that perfectly marries our deepest understanding of viral mechanics with a profound desire to heal. We have tamed the ghost in the machine and taught it to fix the code instead of just rewriting it. So, where does this leave us? We have journeyed from a single virus infecting a single cell to the grand sweep of evolution, to the cutting edge of medicine. And we arrive at a place of quiet contemplation, a place of profound questions about who we are. The central theme of our story has been the blurring of a boundary. The line between self and other, between human and virus, is not a sharp, clear line. It is a porous, shifting, and deeply intertwined border. Your genome, your personal book of life, is not a solo-authored work. It is a collaboration. It has been edited, annotated, and added to by countless viral visitors over millions of years. This viral DNA is not something you acquired during your lifetime. It was passed down to you from your mother and father, who received it from their parents in an unbroken chain stretching back to a time before we were even human. It is as much a part of your genetic inheritance as any other gene. Every person on this planet carries this fossil record within them. We are all, in a very real sense, part chimeras. Our identity is a mosaic of different evolutionary histories. This challenges our very concept of genetic purity. There is no such thing. We are living ecosystems, walking archives of ancient life. And even today, the story is not over. There are more recent viral insertions, the so-called young ERVs, that are not present in all people. Some of us have certain viral insertions that others do not. This means that some of the genetic variation that makes us unique individuals is due to our different inherited viral histories. The fact that you and I might respond differently to a medicine or have a different susceptibility to a certain disease could, in some small way, be influenced by the different viral ghosts we carry in our DNA. So, take a moment to just be with this idea. The quiet, rhythmic process of life that is happening inside you right now, in every one of your trillions of cells, is running on a code that is part ancient primate, part ancient virus. The boundary is blurred. The neat categories we use to make sense of the world fall apart when we look this closely. It's an eerie thought, but it can also be a deeply comforting one. It connects us to the entire web of life in a way we might not have expected. It tells us that we are not separate from the world, but are a living, breathing record of it. The history of life on this planet is not just around us. It is quite literally within us. As we drift toward the end of our journey, let's cast our gaze forward. Where does this strange and wonderful science go from here? What questions are the scientists of today and tomorrow trying to answer? The field of ERV research is exploding with new discoveries. We have only just begun to scratch the surface of what this 8% of our genome is doing. One of the biggest questions is about reactivation. We know ERVs can be reawakened in disease states, but could they also be reactivated by other things? By stress, by aging, by environmental factors? Some researchers are investigating whether the slow reactivation of these viral elements over our lifespan could contribute to the general decline and inflammation that we associate with getting older. Could the ghosts of our past play a role in how we age? The link to neurological and psychiatric conditions is another fascinating frontier. We've already met the ARC gene, the domesticated virus that helps us form memories. Are there other ERVs that influence our brains? Some studies have found tentative links between the activity of certain ERVs and conditions like schizophrenia and ALS. The brain is the most complex organ, and its regulation is exquisitely sensitive. It's plausible that these potent viral regulatory switches, if woken up at the wrong time, could have profound effects on our thoughts and behavior. This is still highly speculative, but it's an active and exciting area of research. Of course, the most powerful new tool in this field is CRISPR gene editing. For the first time, we have a technology that allows us to go into the DNA and make precise changes. This opens up incredible possibilities for research. Scientists can now go into cells and specifically switch on an ERV to see what it does. They can delete one to see what happens when it's gone. This allows us to move from simply observing correlations to proving cause and effect. In the future, could CRISPR be used therapeutically? Could we one day develop a therapy that seeks out and deletes the proviral DNA of HIV from every infected cell, offering a true cure? Could we identify a problematic ERV that contributes to a disease like multiple sclerosis and design a therapy to permanently silence it? These are no longer science fiction questions. They are questions that scientists are working on in labs right now. And perhaps the most profound question of all remains: how much of our humanity is a viral gift? We know about the placenta. We know about the memory gene ARC. What else is there? Did retroviruses play a role in the evolution of our complex brain? Our consciousness? Our creativity? The answer is, we don't know yet. But it's clear that the relationship between our ancestors and these viruses was not just a story of conflict. It was a story of co-creation. We are who we are today, in part because of these ancient, persistent, and transformative viral invaders. So as you finally let go and drift into sleep, hold on to this sense of wonder. The universe within you is as vast and mysterious as the universe outside. It contains multitudes. It contains the stories of your parents and their parents, and it contains the faint genetic echoes of viruses that thrived in a world we can only imagine. You are a library, and every night your cells quietly go about their work, reading from this incredible collaborative book. A book written in the ink of stars and evolution and the quiet, persistent code of the virus that rewrote your DNA to sleep well.